Hi guys, I realize it's asking a lot to ask you to keep hanging in there, but keep hanging in there. I've got some ideas that I think might help a lot. We're going to look at chapter 16 now. Didn't know about the structure of DNA until 1953 when Watson and Crick did the work on that, and we'll talk more about that. And you've also been watching videos with Lab. Griffith is the first one in 1928 who proved that DNA can transform or change bacteria. This was some really interesting studies with Streptococcus pneumoniae. So there were two strains that he was working with. One of them was called the S strain, and it was pathogenic. In other words, it could actually cause pneumonia. One of them was harmless, and even though it was a strain of the same bacteria, it was different enough genetically that it could not cause a disease, and Griffith called that the R strain. So follow what he did. He injected the bacterial cells into healthy mice. Excuse me. Now, mice have been a really good test subject because they're mammals. So even though they're much smaller than we are, they have a lot of systems that are similar to ours. Now, let's talk about the results that he got. If he took living S cells, those were the bacteria that had the ability to cause the disease. They would be called the pathogenic control. Excuse me. Now, if you inject those into healthy mice, what would you expect? You're exactly right. You should get a dead mouse, and he did. And then Griffith took living R cells. And remember, R is the strain that is not supposed to cause the disease. And so this was called the non-pathogenic control, or the non-disease control, the non-disease causing control. So what would you expect to have happened when you inject that into mice? Exactly. You should have a healthy mouse. So even though you have injected him with a bacterium, this bacterium itself doesn't have the ability to cause a disease. Now, then Griffith took S cells and he killed them with heat. So he took cells that should have been pathogenic, actually killed them, and because they were dead, he made them non-pathogenic. And when he injected them into the mice, just as you might expect, the mice remained healthy. Now, here's something really interesting. Griffith then took a mixture of heat-killed S cells. And remember, if you heat kill them, you have treated them, so they're no longer pathogenic. And he mixed them with living R cells, and those were non-pathogenic. And lo and behold, what do you get? A dead mouse, which hardly seems to make sense. But remember, from this dead mouse, you can actually culture live S cells. Okay, now wait a minute. This is really weird. You've killed the S cells. You've mixed them with living R cells. You would expect no disease, and yet something has happened between the two of those strains so that when it's injected into the mouse, it kills the mouse, and you can retrieve live S cells. So what that tells us is something is happening in the DNA that is converting it, transforming it. This is what we call a transformation, a change in the genotype and phenotype due to assimilation. That means taking up and then incorporating foreign DNA. So somehow between these two strains, we incorporated the DNA that transformed the non-pathogenic to pathogenic. Avery McCarty, and McLeod, some of the most important individuals you'll ever hear about, identified this transforming substance. Now, this is so fascinating to me, this early history of DNA, because for so very long, we had absolutely no clue what determines heredity, what kind of substance could be causing transformation. And these guys identified it as DNA, which was very, very important. Specific viruses called bacteriophages or phages, some people pronounce that phage, these attack bacteria. Now, remember, a virus is non-living. It does not have all of the characteristics of living things because it is a genetic parasite. In other words, to reproduce, it has to go in, take over the machinery of the cell, and then reproduce itself using the host cell machinery. It possesses typically a protein coat and a nucleic acid. And interestingly enough, that nucleic acid in a virus can be DNA or RNA. And here's something really weird. That DNA can be single-stranded or double-stranded, and that RNA can be single-stranded or double-stranded. Okay, so go to figure 16. I've got figure 16, 163, 16.3. Find that for me. I don't know what the heck I was talking about. Or it could be, 16.3. Dang, crazy. All right, finally, in 1952, two other investigators did some really pivotal work, Hershey and Chase. And what they showed, that DNA is definitively the genetic material of a T2 phage that attacks a bacterium called E. coli. So here's what they did. Because see, the big debate was, is it a protein that is responsible for heredity, or is it DNA? And what they did is they tagged the protein using radioactive sulfur, which was brilliant because the DNA does not have the sulfur. And then because phosphorus is a very important part of DNA, they use radioactive phosphorus to tag the DNA. So they had a tag for both of these. Okay, so picture this, if you will. Let's look at it. I'm going to draw a bacterial cell, which could look just like a rectangle. And then here's the DNA. And if you look at the virus that lands on it, and what we found out is it injected the genetic material. Now, how did we know that? Because all the radioactive S was on the outside, all the radioactive P was on the inside, which meant that the DNA from the virus moved in and this protein coat stayed out. It was definitely a DNA material.